This is the second part of cell division, and in this we'll talk about types of cell division. So last time we studied that when a cell basically it undergoes a cell cycle when it has to divide itself, right? So there are two phases of cell division. One is interphase and another one is actual division. In the interphase, we said that the cell prepare all the material that it has to use while dividing itself, right? So after the preparation of that material, it enters the actual phase of division, and after that it undergoes the following changes that we'll study in this part. So basically cell division is of two types. One is mitosis, another one is meiosis. We'll deal with mitosis first. Okay, so what happens in mitosis is that one cell divides to form two cells. Two cells are formed from one single cell, and the number of chromosomes which are present in this parent cell will remain same in the daughter cells. So the one which divides is the parent cell, and the two new cells which are formed, they are the daughter cells. So they're trying to say that parent cell divides itself into two daughter cells, and the number of chromosomes, let us say if we talk about humans, we have 46 number of chromosomes. So if a parent is having 46 number of chromosomes, then it means the two cells which it is splitting into will also contain 46 and 46 chromosomes, right? How this is happening? Because we said earlier that during the interphase, this DNA material will be doubled. So it will be 46, 46. And then these two 46 will split into two new cells, right? So mitosis, in case of mitosis, this is also known as equational division, because in this case, the number of chromosomes which are present in the parent cell are remaining same in case of the daughter cells, right? Next, now, even the mitosis is further, it happens in two phases. One is karyokinesis and another one is cytokinesis. So in the first phase, the nucleus divides. Karyo is nucleus, kinesis is division. So in the first phase, the nucleus divides itself into two, and after that the cell splits into two. Now this karyokinesis is also having further phases. Prophase, then it is metaphase, then anaphase, and last one is telophase. So nucleus divides itself into two in the following steps: prophase, metaphase, anaphase, telophase. And once the nucleus is divided, then the cell divides, and this is known as cytokinesis. Let us study this in detail. So when mitosis starts, the very first step is prophase. Okay, so during the interphase, we said that the DNA and the nuclear material is all set, all prepared. After that, this DNA, this chromatin, will start condensing itself, and now it will look like chromosomes. So what happens in prophase is that this chromatin, it starts looking like chromosome. It becomes distinct. Now we can see them clearly under the microscope, right? So even the prophase is one early prophase and late prophase. What is happening in early prophase is that the chromatid is now taking the shape of chromosomes. The chromosomes are becoming distinct, right? And another thing that starts happening is the appearance of centrioles. Centrioles are only present in the animal cells, remember that. Centrioles also start appearing. While if we talk about plant cell, there are no such centrioles, okay. Now what happens in late prophase? In late prophase, this nuclear membrane, nucleus is covered by a nuclear membrane inside which all the material is present. So in late prophase, this nuclear membrane will start disappearing because nucleus has to split. So its membrane will start disappearing. Okay, inside it are present the chromosomes, and the centrioles which were earlier here, they were trying to move on the opposite ends. Now the centrioles will reach the two opposite ends in case of late prophase. Got it? Same will happen in the plant cell, right? So this is prophase. What is happening in prophase is that the centrioles start moving towards the opposite poles, chromosomes start becoming distinct, then chromosomes they are present inside the nucleus and the nuclear membrane starts disappearing. Now the next phase, what was after the prophase? It is metaphase. Now in metaphase, the chromosomes will arrange themselves at the equator. Chromosomes will, you will not draw this nuclear membrane now because the nuclear membrane has become completely disappeared, right? So the chromosomes, they will arrange themselves on the equator. They will all lie on the equator in case of metaphase. Alright, and the centrioles, remember the centrioles which reached on the opposite poles, from these centrioles start arising the spindle fibers. What are spindle fibers? Just the threads. The threads which will act as a basis of attachment. These chromosomes will attach themselves on these spindle fibers, okay, and the chromosomes, they will all lie on the equatorial plane in case of metaphase. Okay, chromosomes are now at the equator. Now what happens in anaphase? Now anaphase, and they are also split into two, early anaphase and late anaphase. So what happens in anaphase is that the chromosomes, these were two sister chromatids, they were attached at a central region known as centromere. Now they will separate themselves, they'll split. The two sister chromatids, they'll split now. So what happens in anaphase is that the chromosomes, they will split themselves and they'll attach themselves to the spindles and they will start moving towards the opposite poles. They'll start moving towards the opposite poles. And what will happen in late anaphase? In late anaphase, these chromosomes will reach on the opposite poles. In early anaphase, they starts moving towards the opposite poles, while in late anaphase, they reach at the opposite poles. Now look at this thing, the nucleus has split into two, right? And apart from that, there starts appearing a furrow. This is a cell. On the cell, on the outskirts of the cell, on the outer region of the cell, a furrow starts appearing. Okay, this means that now the cell is ready to split. The nucleus is almost already split up. Now it's turn of cell, right? So the last phase, prophase we did. The nuclear membrane starts disappearing, the chromosome starts becoming distinct. Then metaphase, the chromosomes, they all lie on the same plane. In case of metaphase, they lie on the equator. And then in anaphase, the chromosomes, they split up and they start moving on the opposite ends. And at the end of the anaphase, they have already reached the opposite poles. Now the last one, telophase. What will happen in telophase is that the chromosomes which were almost there at the opposite poles in late anaphase, now those chromosomes, they'll form their own nuclear membranes and Now the chromosome will again take the shape of chromatin, the thread-like material. So the two sets of daughter chromosomes, they reach the opposite poles, then spindle fibers disappear, okay, and nuclear membrane is again formed, and this furrow, it starts deepening itself. This furrow is furthermore deepened in case of telophase. So now we can see clear cut two nucleus. Now what is the next step? Now the cell will divide, and when the cell will split into two, that is known as cytokinesis. So firstly it was karyokinesis when the nucleus was splitting into two, and that happened in 4 steps: prophase, metaphase, anaphase and telophase. And at the end, due to this furrow formation, the two cells, they split up, and this is known as cytokinesis. So almost same was going on in the plant cell simultaneously, but there were some differences in animal and plant cell. In case of animal cell, we saw the formation of furrow, right? But in case of plant cell, this furrow is not formed. In case of plant cell, there is formation of plate. So the cell divides into two by the plate, okay? And you can also see that centrioles were present only in the animal cells. There are no such centrioles in plant cell. So even these organelles, centriole are the organelles. They are only present in the animal cell. They are not present in the plant cell, right? Yeah, that's all about mitosis, right? And what is significance of mitosis? Mitosis is helpful for growth. It is helpful for this repair of the cells, then replacement of the cells, and then in case of asexual reproduction also, mitosis is taking place, right? And what about meiosis? Meiosis is not as such in your syllabus, but still what happens in meiosis is that meiosis is known as reductional division. Reductional division because in case of meiosis, one cell splits into 4 cells, and if the parent cell is having 46 chromosomes, let us assume, then the cells, the daughter cells that will be formed at the end will be having half the number of chromosomes, let us say 23 each. So if the DNA gets doubled, let us say 46, the double of 46 is 92, right? So 92 is splitting here into 4 parts. 23 into 4 also gives you 92. Okay, so one cell is splitting into 4 cells, and they are having half the number of chromosomes as compared to the parent cell. So that is why meiosis is known as reductional division, right? Because the chromosomes are becoming half. Okay, this generally meiosis only takes place while the formation of gametes in case of sexual reproduction. Because what will happen if we say that while the formation of zygote, what happens during the formation of zygote is that a male gamete fuses with a female gamete, right? The male gamete, it fuses with the female gamete, and then a new baby, a zygote, is formed. So if the cells will already be having 46, then the baby will be having 92 chromosomes. That is quite abnormal situation. So while the gamete formation takes place, one gamete should consist of half the number of chromosomes only, so that 23 of male, 23 of female, they combine together and they give rise to individual having 46 number of chromosomes, which is quite a normal thing, right? So what happens in meiosis is that firstly one cell divides into 2. Okay, if this cell is having 46, then it will divide into 2 cells having 23 and 23. After that, these 2 cells will undergo mitosis. What happens in mitosis? The number of chromosomes of the parent cell remains same in the children, in the daughter cells. So this 23, it will double itself, and then it will split into 2 with 23, 23, again 23, 23. Right? Firstly, one cell splits into 2 and the chromosome number is halved. half of 46 is 23. Then again these two cells divide into further 2 more cells, and hence one cell is giving 4 cells with half the number of chromosomes. Okay. So one thing is clear, that how is zygote formed? When one male gamete and one female gamete, they'll fuse, they'll form a zygote. Now we say that, for example, some of our characters, they match with our mother, some of our characters, they match with our father, and us as individual, we are also having some of the distinct features which do not match with our parents, which do not resemble with our parents, right? How are they being appearing, or how are they being so, visible, or how are such characters possible, right? Because we are formed out of the genes of our mother and father. So the genes are basically kind of mixing up, right? But still we are having some of the things, some of the features, some of the things that do not match with our parents. So how is this possible? He says that now when this chromosomes of, let us say, maternal and paternal, while this maternal and paternal chromosomes, they also exchange their materials, like this white, let us assume this is white chromosome, this is black chromosome. Before formation of zygote, even these chromosomes among themselves, they share their parts with each other. For example, this white will share one part and this black will give its part to this chromosome. So this is known as crossing over. Okay, while the maternal and paternal chromosomes are separating, their material gets exchanged between the two members of the homologous pair. These are the chromosomes of, let us say, male father. These are the chromosomes of female, right? So the chromosomes of one parent, they exchange their materials. Similarly, exchange of material occurs in the chromosomes of a female. So even before the formation of zygote, this exchange of material is taking place. This is known as crossing over, and due to this crossing over, we are having some of the features which are different from both our parents, right? So this is all about your chapter. Make sure you practice all these things. Make sure you know each and every stage. Question will be asked in such a form that, for example, they'll give you a picture. They'll place the chromosomes on any of the, it can be prophase, it can be anaphase, telophase, it can be whatsoever. They'll give you a picture. They'll say, name this stage of mitosis. So if they are lying on the equatorial plane, we'll say this is metaphase. Now the next question will be, next subpart will be, which state will come prior to this? Which stage will come before this metaphase? Then you will say prophase. Then another question will be, which stage will come post metaphase, post this stage? Then you will say anaphase, right? So they'll give you an image, they'll ask you question related to that which stage is this, what will happen next, what stage will come next, and what happens during this stage, etc., etc. Or they will mention a name, they'll say anaphase, and they'll ask you what happens in anaphase, right? So the questions can be asked in these two forms. Okay, all you need is a lot of practice and you will be able to do it.